This tiny drop of liquid can kill a human in minutes. One bite, one mistake, and there is no second chance. Experts extract this deadly venom. Yet, inside this laboratory, every single day to save human life. A group of team entering the jungle to locate venomous snakes. They are carefully searching for the targeting snakes. Spotting the snake without disturbing it is crucial.
Preparing to secure the snake's head safely.
Snake captured and placed safely for transport.
Okay, Ahatula, looks good here. Copy that. This nausea is secure. Good. Let's check the next one.
Multiple snakes collected, each carefully labeled. Venom is the key ingredient for life-saving antivenom.
Here we see the venom proteins from the species in this crate. A holographic model shows how they bind to receptors. The biohazard alerts highlight the high-risk nature of this research.
Bringing snakes safely into the extraction lab. Venom is carefully collected drop by drop. Precise measurement ensures safe antivenom potency. Vials are labeled and organized for processing. Venom proteins are analyzed at microscopic level. Science ensures every drop is effective and safe. Purifying venom proteins for safe antivenom production. Industrial lab processes concentrate venom proteins. Purified venom is collected into labeled vials. infographic shows raw venom transforming into purified protein. Scientist inspects purified venom under microscope. Purified proteins are ready for next stage. Horses generate antibodies, the foundation of anti-venom.
This batch is potent. Draw slowly.
Understood? I'll prep the next vial.
Precision is key here.
Agreed. This will yield good data.
Exactly. Let's finish this series.
Small doses of venom are injected safely into horses.
Easy now, big guy. This won't take long. All done. You did great. Now for the sample.
Blood is collected and labeled carefully.
In horse blood, antibodies form as Y-shaped proteins bind to antigens. This triggers the immune response, protecting the animal. Researchers analyze this data to develop new treatments.
Scientist monitors antibody levels digitally. Antibodies are ready for antivenom extraction. Purified antibodies become life-saving antivenom.
Flow rate is optimal, initiating separation protocol.
Confirmed. Monitoring for hemolysis.
Antibodies are extracted from horse blood. Purification panel ensures proper concentration. Vials are organized with batch numbers.
From blood, antibodies are extracted. These are purified to create the antivenom. A final check ensures the antivenom is ready.
Scientist inspects antivenom under microscope for quality. Each batch is tested for safety and effectiveness. Scientists examine antivenom vials under microscope. Assay plates show potency results. Digital metrics track dosage and concentration. QC passed stamps are applied on batches.
Antivenom undergoes rigorous safety checks, including purity testing to ensure it is free from contaminants. Finally, tests like the protopalm assay verify overall safety. This ensures every batch is safe and effective.
Vials are ready for shipment after final inspection. Packaged safely and shipped worldwide. Antivenom vials are packed into boxes. Labeling machine prints batch numbers. Boxes are loaded into refrigerated trucks. Global map overlay shows shipping routes. Anti-shock foam ensures safe transit. Trucks are ready for dispatch to hospitals.
Newton and Einstein formulas are applied to highlight scientific principles. Patients receive life-saving doses. The process from venom to anti-venom saves lives globally.